So now the most important is gene therapy. So in the field of the biotechnology, so gene therapy is the process where the genes are inserted or removed. So desired gene might be inserted or removed. So this process called gene therapy. So gene therapy is not an easy task. It is just a insertion of the DNA into the nucleus. It is a very complex process. So first gene therapy occurs in the patient of SCID. SCID means severe combined immunodeficiency patient or sickle cell anemia. So first gene therapy takes place in the 4 years old girls who is suffering from the adenosine deaminase deficiency, is called ADA deficiencies. So this process, this therapy occurs in first time in 4 years of girl. So ADA deficiency. So this is the process, the procedure method how the therapy takes place. First we remove the desired gene by the process through the action of the nuclease enzyme and synthetic enzyme, DNA synthetic enzyme, and produce the cDNA. cDNA produced by the reverse transcription method. It is a single stranded DNA. It is a single stranded cDNA. So complementary DNA. So complementary DNA which transmitted through the virus. So SV virus which act as a carrier or vector and carries this cDNA of ADA gene, cDNA of ADA gene, and then the cells removed from the lymphocyte cells. So cDNA inserted into the lymphocyte by the viruses. So then the lymphocyte which contain cDNA or DNA of gene of ADA gene, ADA gene, and then it finally inserted into the bone marrow cells. It is inserted into the bone marrow cells, and then bone marrow pluripotent cells which produce this enzyme will be produced through this process. So this is the gene therapy method. So this is all about. So first, and another process is called hybridomas technology. So hybridomas technology is not a DNA recombinant technology. It is just a process of the complete fusion of cells, complete fusion of the two cells. So here is the product. So in hybridomas technology, production of monoclonal antibody takes place. Production of monoclonal antibodies takes place. So how the monoclonal antibody produced? So this process first is the B lymphocyte. So B lymphocyte is the main function of B lymphocyte to produce the antibody, and another cells is myeloma cells, is a type of cancerous cell. The main function of cancer cells to divide rapidly. So rapidly divide cells and antibody producing cells. So this is the combination of both cells in culture medium. So through here is the parasexual fusion takes place. This is the parasexual fusion, means complete fusion of the two different cells. So this is the parasexual fusion, and parasexual fusion formed a cells. That cells is called hybridomas cell. So hybridomas cells have a ability to produce antibody as well as rapidly divide. So rapidly divide cells which produce more antibody. That antibody is called monoclonal antibody. So monoclonal antibody acts on the specific gene. So it also helps in the many diagnosis process. This is the main fast growing and produce more and more antibodies. So this is all about related to the biotechnology and application of the biotechnology in the field of medicines. So more and more medicinal product produced through the recombinant DNA process and complete fusion as well as gene therapy process. So this is all about related to the biotechnology. Next we will discuss application in the agriculture field. So, agriculture, so biotechnology in agriculture we will discuss later or next class we will discuss. Okay, thank you.